Okay, so I think we might as well wrap up. I think we have scheduled one last lecture, and I think it's probably been a long day, 2 days for everyone, so I'll try to be brief, but also to try to put together a bunch of things that we've been talking about over the last couple days. Let me just go dive into it. So what I want to talk about in the last maybe 20 minutes or so is talk about how we've been using these tools and where I think these things will be going. So the first area to think about is this graph that I showed last time, which is a graph of publications from our lab with the year of publications versus the time scale. And it's interesting to sort of think about projecting this exponential further. And we have a work from JHE and others in my lab that's going to be describing timescales that are much longer than 10 milliseconds. And as we go further out, I think this technology will be continuing to go further and further. We can debate when the first timescales on the orders of seconds will be reached, but I don't think this will take 10 years to do. I think this will be maybe 5 years or a few years. And the exciting thing about that is that once we're talking about seconds, ironically, the jump from seconds to minutes isn't that much. That's only a factor of 60. I mean, milliseconds to seconds is actually a big deal. Seconds to minutes is pretty trivial in comparison. And seconds to hours is more like milliseconds to seconds. And so actually now we're starting to talk about timescales where really we don't worry too much about the timescales at all. And it's kind of interesting to think about MD because normally when we first think about MD, the first thing that we think about is, oh, can we reach the timescale? And to get to the point where we can handle such long timescales such that we can stop worrying about that, that would be a pretty dramatic change. So we're obviously not there right now, but I don't think it's necessarily that far away. For those of you that are, let's say, first-year grad students, by the time you're finishing maybe your PhD or your postdoc, with a timescale where we're starting to get into, I think, this range, maybe 5 or 10 years from now. And that actually, I think, is an exciting thing. One thing that I think you've also seen with Liping Wang's work on force balance is that hopefully, relatively soon, we won't be just talking about getting to long timescales also with force fields that are considerably higher level accuracy. And I think that's also something that will be very exciting when combined. To maybe put some sense of where we're going and what we've done with this and where I think the future of this lies, it's useful to talk about where we've been. And so one thing to talk about is, for instance, we've done a lot of work with protein folding. And I think protein folding is interesting because it's often a canonical problem for many other fields. Actually, when I first came to Stanford, I was chatting a lot with Michael Levitt, who's been a great mentor of mine. And one thing that Michael said very early is that his interest in protein folding was especially in the fact that it's such a great paradigm for other problems. That if you can tackle types of problems involved in protein folding, you can tackle a wide range of other things. I would add one thing to that, which is that actually the types of phenomena we see in protein folding, we're starting to see in other areas. And I think in the vignettes I'm going to talk about in the next 20 minutes or so, I think we'll see some examples of that. So first off, is that one thing that we see now that we have long enough timescales to reach it, and methods that really don't bias you, don't force you towards certain outcomes, is that we see the prevalence of a lot of non-native interactions. So we see the prevalence of structure that isn't found in the native state. Or if we're talking about conformational change from A to B, we find structure that's in neither A nor B. And in path-based methods where you pull from A to B, often this doesn't happen. In those methods, you typically break apart the stuff in A that's not in B, and then put the stuff in B that's not in A together. And in the movie that Liping showed for NTL9, we saw these steps where at each one of these steps, the protein was until the very end, had a series of non-native structure that wasn't present in the final structure. And so that alone, I think, is something that was going to start becoming more and more prevalent and something that there's hints for experimentally, but it's an area where theory and computation really can lead. Because if we see these things, we can make very specific predictions of where to put probes or what mutants to do. And so one other reason for bringing this up, I think, is that one area for the future of MD, which I think is particularly important, I want to encourage all of you to think about, is not just running MD for the sake of sort of giving a posteriori justifications for things, but using in ways that we can lead experiments and make specific predictions for what experiments should be doing. Now, naturally, not all those predictions will be right, but often we can be right with a relatively low frequency and still be very useful. If you make 5 predictions and 2 of the predictions turn out to be interesting, that can be really quite powerful. The reason why is that imagine someone studying a relatively small protein, like a 100 residue protein, and they want to come up with ideas for where to put probes. there's roughly 10,000 or 100 squared locations for these probes. And if you do it randomly, you probably won't find things that are particularly interesting. And whereas hopefully computation can do much better than that. Okay, so then the other point that, that comes up that we see in protein folding a lot, that seems to be very common, is this ubiquity of having many paths and many states. And this is a tricky thing because I think part of the reason why we see so many paths and so many states is the fact that we have methods now sensitive enough to see it. And so this is an example of a transition path theory diagram for NTL9, where we're going from an unfolded state to a folded state. And this is a lumping diagram with just a few coarse-grained states. But even still, what we find is while there's one path that dominates, there's many other paths that exist. So this is also an interesting point for tension with experimentalists, because often experimentalists would maybe do an experiment on this system and say there's a single path. And that's often because their signal to noise would find this dominant path, but really wouldn't be able to see other paths. And so, and maybe that's for that particular system, if one path maybe is present in 70% of the time, then that's the natural one to talk about. And you can ask, well, why would even bother with these other paths or these other states? Well, one thing to think about is that often these other paths or the other states, even though they seem to be low probability, low is a relative thing. So even if the probability is, let's say, 10% or 1%, it doesn't take many kcals of energy to turn something from 1% to 99%. It only takes a little more than almost 3 kcals to do that. And so the fact that even the 1% are low in probability is not really it's an important thing in the sense that it doesn't take much in terms of mutation of small molecule to make them important. And that's another area where theory could potentially be making some very important predictions. And again, hopefully leading experimentalists into interesting areas. A side note is that when we're doing all this, we have to be able to hopefully make these connections to experiments. And one of the things that we and many others, and I think many people in the room, have been trying to push forward is trying to make very detailed connections with experiments. And so one of the benefits of MSMs and one of our real motivations is that we want to not just be able to show movies, but really make quantitative predictions very accurately. And I think that's something that's going to be very important for us to be making connections and making a huge impact in any given field, is to be able to make predictions quantitatively and then directly compare them with experiments. Okay, so the third and final point about protein folding, which I think is going to be a general point, and we'll see in other areas, is that now once we have this MSM formalism, we can actually start to ask interesting questions that you couldn't easily ask before. So in the folding sense, one thing that we asked is, what is the network connectivity? So how is the native state connected to various non-native states? And one of the things that we and others have seen is that the native state is what's called a kinetic hub, that it's very highly connected, that there's many paths into the native state. And so this is, I think, an interesting work in terms of its impact for protein folding. But I think there's a more general statement here, even for those of you that aren't interested in studying protein folding, which is that once you have the master equation, you can ask interesting questions of it that you couldn't easily ask of just a straight trajectory. You can ask questions such as, what is the nature of the network topology? And actually a variety of other types of questions that really the master equation really facilitates. And so if you guys are going to go through all the effort to build MSMs and to make these quantitative predictions and maybe visualize things, I think there's all this richness that's sitting there in terms of understanding it afterwards. Then finally, one other thing to think about, and I don't have a slide for this, but it's related to this question, is that once you have the MSM, you actually have a very powerful tool, because you can generate many trajectories very easily from that. In a sense, you have something that has the capability of what usually people associate with toy models. That people like little simple lattice models or analytic models because they're very quick to run. But once you've built the MSM itself becomes a very powerful, easy to run model. But unlike these toy models, your MSM hopefully can quantitatively predict experiment. So the ability to have a toy model in facility that has experimental fidelity is really kind of an amazing thing and a unique opportunity. Okay, so those are some thoughts that comes from protein folding. Let me give you an example for why these ideas in protein folding might be relevant for you by talking about examples very different from protein folding. And so the first example is talking about kinases. And so kinases is something that we have been ramping up in our lab, and I know there are people here from John Curran's lab that, and others that are very interested in kinases. One of the driving questions for understanding kinases, and especially how kinases behave, is kinase selectivity. That what we're looking at here is graphs of the kinome, this family tree of kinases, with the places where a drug would interact superimposed. So there are some cases where a drug is relatively specific to just a certain part of the kinome, but there's other examples, and most examples, where the drugs are fairly nonspecific. And having nonspecific drugs is a really nasty thing, because we need our kinases to do things, and kinases are typically cancer therapeutics. They are intended to leave kinases inactive. But if you have all of your kinases inactive, you can imagine that there will be all these nasty side effects. And so one of the goals for us in understanding kinases is to try and understand whether there may be some hope for coming up with drugs that might be more selective. So towards that end, one thing that is natural to do is to just study the mechanism of activation. And instead of inhibiting kinase activity by locking you into the inactive state, maybe there's something along the way that you can stop that would prevent it from becoming active. Actually, interestingly, the kinase field's also now becoming interested in kinase activators for other types of therapy. And so the same could be true, that by understanding how you go from inactive to active, you can maybe also come up with new ways to activate the kinase. Okay, for those of you that aren't experts in kinases, let me just walk you through what the difference is between inactive and active. And kinases, I think, are interesting in terms of conformational change, because unlike other systems like GPCRs, the conformational change is maybe a little more easy to see with your eye. So there's some examples of particular hydrogen bonds that break, or particular hydrogen bonds that form, or particular hydrogen bonds that break. Also, there's an activation loop that goes from essentially something that is kind of, has some folded-like structure or some well-defined structure to something that is more unstructured. And then finally, also, there's a helix here that rotates. And one question is, how does all this happen? Does it happen step by step, or does it happen in some concerted way? And in particular, one of the things that we've been interested in when we look at this is whether there may be intermediate states that might be interesting targets. And so when we look at dynamics, and this is dynamics from a MSM movie, so we took snapshots from the MSM doing this kinetic Monte Carlo procedure I described yesterday. One thing that we see is that when we compare the trajectory to a projection to the degrees of freedom I just talked about, whether these hydrogen bonds are formed or whether the A loop is formed, one thing that we see is that the system tends to hang out very much in two intermediate states. And actually, the presence of the intermediate states were actually kind of interesting, an interesting surprise, and maybe suggests that they could be interesting allosteric targets. And this idea of having an allosteric target now is actually becoming very exciting. And there's many other systems for which people are hoping to be able to target systems in this manner. The idea of an allosteric target is that we want to find some site on the protein that's not the active site, because the active site might be very similar to other sites. In the case of kinases, the active site involves ATP binding, which is pretty generic, whereas these allosteric sites may have the hope of being much less generic. Okay, so we can run the simulations and we can make some intriguing hypotheses, but the next step is to try to make connections to experiments, and that's something that's still ongoing. But in the case of this particular kinase, one thing that was cute is that Diwakar Shukla, the postdoc who's really driving this work, compared his intermediate structure to a structure of a related kinase from the PDB, where this related kinase has a drug already bound to it. And actually, the crystal structure for this other kinase, CDK2, is actually very similar in structure to the behavior of the intermediate. And so while this is by no means proof, this is at least sort of intriguing that we might start to be able to start to experimentally see these kinases by binding specific drugs and then co-crystallizing. And this is an example of something that I was talking about, where there's the hope that by doing these simulations we can make specific predictions and guide experiments in specific directions. So one other area that I think is very powerful in terms of post-analysis, and I think, I don't know how much we've emphasized it here, but as building MSMs becomes easier and easier, I think the new challenge switches towards understanding what the MSM tells us. And Munib and others in my lab have been working, and Christian, who's here too, have been working on methods in these areas. And one area that's been useful is looking at mutual information for correlations. And so this is an example where I think often understanding correlations is useful. For a lot of people, when they look at this picture, they have a hard time guessing what it is. But if they could see the correlated dynamics, they usually have a much easier time seeing what it is. And What this is a four-stroke engine. But the idea is that if you just look at a snapshot of the engine, maybe when the car isn't moving, it's hard to understand its function and its dynamics. But by understanding the correlated motions, you can identify that there's specific parts. You can see that there's pistons. And you can also see that there's correlated motion. And the correlated motion can give you a lot of idea for how this thing would function. And so you can imagine doing an analogous thing for proteins. In this case, what we do is that we look at mutual information between the dynamics, something that can be driven straight out of the MSM. And in this case, we can see that certain regions of the protein are correlated with each other into various blocks. And in a sense, these blocks define the parts. And what's powerful about this is that, this is a 200-ish residue protein that if we did it residue by residue, that's too many parts, I think, for us to put into our head to try to figure out what's going on. But if we can break it down into fewer parts, I think we can, A, understand what's going on, and B, start to make very specific predictions. And I think the nature of the predictions that we can make, and the thing that's tantalizingly interesting about this, is that we find correlations between parts, such as the active site, with parts that are pretty distal to the active site, in various parts of the protein. And this makes very specific predictions for how mutations very far away from the active site could affect function. And mutations such as these have been discovered, and we can and have correlated to those, and I think we're doing pretty well in predicting them. But this is another area where I think computation really can be very useful to make predictions or to help understand things that seem hard to imagine, like how could this thing be all that important when it's so far away from the active site. So the last vignette that I want to talk about is a vignette about GPCRs. And in this case, I want to highlight a couple different issues. One issue I want to highlight is how conformational dynamics can be more directly related to thinking about drug design, but also how I think computation, and especially a structural view, can help take sort of the very first models that biologists would use and add a great deal of sophistication to them. So GPCRs are interesting because they are these extracellular proteins for which small molecules bind, and then they transduce signal to inside the cell. In that sense, and what small molecules do is they can turn them on and turn them off. And often we think of GPCRs, at least the way they're described in, let's say, a biology textbook, is they're described as switches. They're on or they're off. And so, for example, you could compare their activity of the GPCR versus ligand concentration, and agonists would turn them on, antagonists would turn them off. And so this all makes sense, except for the fact that we know experimentally, empirically, that there are other things that can happen. There are small molecules that serve as inverse agonists that can have even negative behavior. And there's small molecules that serve as partial agonists, and partial agonists at different levels. And so the intriguing thing here now, especially as people have been pushing for new structures for GPCRs, is that each of these different functions is probably related to a new structure. And crystallography is very challenging here, and I think there's been a lot of beautiful work that can come up with structures occasionally of antagonists and rarely of agonists, but generally not of other things so far. And this is maybe an opportunity for computation to try to elucidate how the function of this, in terms of whether it's an agonist, partial agonist, antagonist, or inverse agonist, how these functions can be related to their structural properties. And this would also be useful not just for understanding GPCRs, but potentially for giving new insights into designing new drugs. The reason why GPCR is especially interesting for drug design is a third of all known drugs hit some sort of GPCR. So I've got some caffeine here that hits a particular GPCR. Every morning I take a drug for my asthma, and that hits beta-2, the GPCR I'm going to talk about in a second. They are such key drug targets. Okay, so let me tell you what we did and talk about what I think the significance of it is. So this is a picture of a cartoon of the beta-2 simulation where it's sitting in a membrane, and it's this 7-transmembrane helix protein. And I think at the end of the day, especially since this is on a GPCR specialty audience, I'm going to sort of skip some of the details here. But we were able to simulate the system, build an MSM, and one of the things that I think has been powerful for connecting to experimentalists is that there's different ways to represent an MSM. One way, which is potentially actually kind of crude, but I still think powerful in connecting to experimentalists, is that we can build trajectories from the MSM and plot the various features that experimentalists are used to looking at and the community is used to looking at versus time, and show activation in terms of the simulation almost as if it was like a longish single molecule experiment. So longish for simulation, probably shortest for experiments. And through this we can see that this thing is activating and inactivating and other details. I think I'm going to skip the specific details of activation. But here's the intriguing part, which is that we can build an MSM, we can see that it has certain properties, but at 3,000 states it's kind of hard to visualize. I think earlier I mentioned how we would take a look at this in a lower resolution to gain some qualitative insight. And what we see is that we see these intermediate states that might be of interest. And so the real question is, what do these intermediate states tell us? In the kinase case, it gave us some hope that there might be interesting targets. In the case of this GPCR, there are already a lot of small molecules that target this thing. And so we're curious to see if whether these intermediates may actually be relevant for function for these small molecules already. Okay, so how do you test that? Well, one way to test this, or to poke at it computationally, is to look at docking to these small molecules. And so what Morgan Lawrence did in my lab is that she docked many of the typical GPCR drugs to different structures. And without going into all the details, what we find is that the docking to the MSM structures improves your ability to improves your statistical strength in docking. There's a statistical metric called the area under the curve. It's the area under a receiver operator characteristic curve, ROC curve. And the improvement, I think, is relatively modest. I think the improvement goes from 0.75 to 0.8, where 1 is perfect and 0 is random. Sorry, 1/2 is random. But it's something where, it is improvement. And actually, even that type of improvement has significance in pharmaceutical companies and something that's gotten interest in pharmaceutical companies. But I think what really got interest was that when we looked at the docking to these intermediates, we found an interesting surprise, which is that if you break down the docking by chemotype, there are certain molecules that only, and in this graph here, this is graphing the MSM states going from inactive to active. And so we saw that there's certain types of chemotypes that only bind to inactive, that they sort of show up here, and only some types of chemotypes that primarily bind to active. But I think the interesting surprise was that there are certain types of classes of small molecules that only bind to these intermediate states in the docking. And so we can't prove that they're binding to these states experimentally without doing co-crystals and all this stuff. But I think the intriguing thing is that, first off, is that we can see that if we didn't have these MSM states, our ability to predict binding to GPCRs would be much worse. And having these states is a huge technological advance, especially in terms of coming up with diversity in small molecules, which is something I think of great interest in pharma. But the more tantalizing thing, which we can't prove, but we can only suggest, is that these intermediate states actually have this purpose. And that when you bind to these states, actually we're creating these sort of partial agonists and having interesting function beyond just being active and inactive. So one cute little anecdote about this is that, and something that I'm particularly proud and excited about, is that after going through this paper, a group at Pfizer was very excited about this work and came up with a job post that, to me, almost looks like the method section of this, of our paper. They laid out that they want people to know about MSMs and a few other techniques that we used in that paper, and actually later talked to them, and it turns out that it's more or less exactly what happened. And so it was very gratifying for me because we've been working on MSM technology now for 10 years. And it takes a while for things to get impact in various areas. I think in the academic area, there's been a lot of interest in MSMs. Seeing this moving into pharma, and I think especially this particular result where it could improve the predictive capability, I think has gotten them quite interested. And I think hopefully that's just the beginning. Okay. So, to conclude, many of the features of the pathways that we saw unfolding, I think are similar in other systems as well. I mean, the common elements in this sort of vignettes that I laid out is that we often see parallel pathways. And I think the MSMs really can give you the power to see these things. Because we're not pulling it from A to B, the MSM can relax in ways to find things that I think other methods can't. Secondly, I think MSMs are pretty good at finding intermediates. That's what this is all about. And in many cases, these intermediate states are the very things that are very difficult to see experimentally. But yet, maybe with the right predictions, can be realized either in terms of co-crystal structures or in terms of the implications for various predictions that could be made, like you make this mutant or you bind the small molecule. And finally, the non-native interaction part is particularly intriguing because it's very difficult to see in a very sort of standard mindset where you have two crystal structures and you want to morph from one to the other. And I think the ability to see these non-native structures, I think, is very intriguing. And we've seen it now in several systems. We'll see how common it is. Okay, so with that said, let me just take 2 more slides to talk about where we're going. In a sense, I meant this to be the future of MD, and I think what we've done just in the last 6 months is still quite challenging. We, in the case of the GPCRs and the kinases, threw a lot of resources at this, resources that most labs don't have access to. But I think maybe in a year or 2 or 3, what we've done here would start to become much more common. Running 1,000 GPUs for 6 months right now is a large calculation. I think in 3 years, it's not going to be a huge calculation at all. And so in that sense, the stuff that we've started doing here is I think, going to be the future of the way other people will do things when the resources become less extreme. But maybe it's interesting to sort of push even further into the future and thinking about what will we all be doing maybe 5 or 10 years from now. And one of the things that I'm personally interested in is in using MSMs to study not just a single protein, but multiple proteins. And the dream is that in short term, we study simple pathways. And so an example of maybe the most trivial pathway, and maybe this is so trivial it's not worth calling it a pathway, but we can look at autophosphorylation, where one kinase phosphorylates the same type of kinase. And the cute thing about this is that, remember, the MSM is just a kinetic model, but it's a very detailed, conformationally aware kinetic model. What we can do is we can build these kinetic models into models of other processes, like, let's say, the autophosphorylation pathway. And what we can do is we can come up with models that now describe much longer timescales. And so even though the molecular relevant timescales here are sort of sub-millisecond, like 500 microseconds, because of the timescales for diffusion and other things, the experimental timescales are typically seconds to minutes. And so what's intriguing about this is that it's a way for us to make even more detailed connections to experiments, but at the same time having a very strong connection to the molecular system. If you compare this to other models that people will make of pathways, they'll have to come up with parameters that come from experiment, rates or other things that come from experiments. Here, everything comes up from the simulations. And what that allows us to do is that, unlike other more systems biology models that come from just experiments, we can say, well, what happens if you make this mutation? Or what happens if you do this, bind this small molecule? In these cases, we could run the simulations and make specific predictions. And so from my point of view, I really care about how the chemical detail affects the cellular behavior. And that's something that we have to hope to do. It's intriguing to think about taking this further, because we start with a trivial pathway, we do a bigger one and a bigger one and a bigger one, and eventually we're starting to talk about really complicated pathways. And P53 is on our minds because it's such an interesting system in many ways, but it's also involved in all these different pathways. In time, and this might take like 20 years, but I think in time we'll be able to describe big complicated networks of pathways and starting to become essentially simple models of how cells behave, but with a strong connection to the atomic and molecular details, such that we can ask, what happens if we study one of my SNPs? How does that behave? Or if Alex comes up with a new drug, how would that change cellular behavior? These are things that current models couldn't do without doing lots of experiments. And I think in the future we'll be able to do this. So 20 years is kind of a hard timescale to predict. My guess of saying these crazy things is based on where we were 20 years ago. So 20 years ago, what are we now? So 20 years ago was 1994. Yeah, 1994. And so I was a grad student on the verge of getting my PhD. And to talk about running simulations of GPCRs and all atom detail on millisecond timescales would just be insane. And at that timescale, at that year in 1994, we could run for a few picoseconds for very little things. But 20 years is a long time, and I think computers will continue to accelerate with Moore's law in terms of doubling of number of transistors for, I think, quite some time. And I think the power is going to be out there to do some very cool things. And I think building upon the stuff that you've seen here, I think we'll be able to do some exciting things in the future. Okay, so with that said, I'd like to thank the members of my lab, many of whom you've interacted here. And thank you all for coming out for the workshop. I hope you got something useful out of it. And it was great to have you come out. Thanks.